Hi, I'm going to give you some instructions on how to do a lab looking at dispersion of a population of a species and using plot samples. And we're also going to go over how to calculate dispersion index through that. I'm Sue Glenn. This is set up for my ecology classes, but I think it's general enough to help you wherever you are. We are using the Ecology Lab Kits, terrestrial ecology kit. And we're also using the textbook by Molles and Sher called Ecology Concepts and Applications, 8th edition. Uh, and then we will be using Excel for doing our calculations. When we're looking at collecting data on a population, the population is defined as the complete set of objects when you're thinking about it statistically, but we often cannot do that. We're trying to do a census in the US and trying to count everyone, and it's obviously very difficult to count every individual, and it's probably harder for organisms that are hiding from us. So in these cases, instead of using the entire population, we use a statistical sample. So we're taking a subset of the population. And when we take a subset of the population, we're assuming that that subsample is representative of the entire population. So if we look at this sample taken from the lake, we're assuming that these different species and these age distributions and these sizes are indicative of what we would find in the overall population. With that, you can make inferences about the characteristics of the population. However, the reliability is going to be dependent on how well the data was collected as to how representative it is of the actual population. So we spend quite a bit of time designing how we're going to do our samples. And sample design is just the method that you're going to use to select the individuals from the sample population. And it's a crucial step to take the time in the sampling process. And there's really two groups of sample design, non-probability and probability designs. Non-probability samples are where you have the individuals are selected by the researcher for specific reasons. So you might be selecting just the wolves that were introduced to Yellowstone from Canada, as opposed to looking at the wolves that have moved across the border from Canada. You might be focusing specifically on the ones that we flew in and established a new population or a replacement population. Instead of selecting particular individuals, probability sample requires you are objectively selecting individuals from the sample. And when you do this, you can determine the probability of selection probabilities and calculate sampling errors. And these allow you to use inferential statistics and the use of probability sampling designs. So when we're using probability samples, we're basically making some assumptions that the percentage of characteristics we see in those samples are representative of the original population, and that we can draw inferences that way. There are different types of probability sampling. There's just simple random sampling where each individual has an equal chance of being selected for a sample. There's a systematic random where you're basically taking every 10th individual or every 5th individual, and you're basically starting from a random spot and then going every 5 trees or every 10 trees or every random number of trees, a systematic system. Or there's a stratified random system. Sometimes you might know that there's different groups in the area that are of interest to you. So you might be making sure you get some of the adults and some of the juveniles, or some of the ones that live, some of the zebras that live in the highlands and some of the zebras that are living around the watering holes. And then you can, once you've divided into those samples, then you can select a random sample from each group. Even though you're picking random samples, not all random samples are the same. They're at random. So here we have on sample H, we have where we've got a very even distribution across different types of soil of the different samples are basically found everywhere. And then here there's some random samples that were based on random coordinates, and we're missing a lot of sampling on this one soil type. So that's sometimes why you would do a stratified sample, is you'll say, well, I want to make sure I get some of my samples here and some of my samples here, so that I have them in the different areas, in the wet areas and the dry areas. So sometimes you want to randomly sample in a stratified way. I have a lecture completely on distribution patterns at small scales and large scales, and I will link those at the end of this video. But in general, when we're talking about patterns of dispersion, we're talking about whether the species, individuals of a population, are found randomly across an area, which means you have an equal chance of finding them anywhere, whether they have a regular, uniformly spaced out distribution where they each have an exclusive territory or an exclusive area where they avoid each other, or whether they have a clump distribution where there's an unequal chance of being anywhere. That might be they might be attracted to each other, or they might be attracted to a certain resource and they're hanging around those resources, or they're hanging out in a herd or a flock or whatever is causing them to be patched. Or it could be the way they're reproducing. You could have a patchy distribution because they're starting from one location and spreading out. When we look at the distributions of individuals in a population in small scales in these different patterns, these patterns are basically coming from different processes within the population. So if we have a random distribution pattern like we have on the left, we're assuming there isn't really a lot of strong interactions causing individuals to be attracted or pushed away from certain areas or each other. Whereas if you have a very evenly spaced out population, like this might be bird nests where they each have a territory and there's antagonistic interactions between individuals, or it could be where they're depleting the resources in an area and no one else can live too close to each other. So it could be desert shrubs that are using up all the water in each individual spot around them. And then clump distributions are also called clustered distributions. They can be called contagious distributions. So this is where you find very high numbers and then areas with very low numbers and then high numbers and then low numbers. So you've got very patchy distribution. And in those cases, they're either attracted to each other or they're attracted to a common resource, or it might be how they're spreading like a contagious disease. In this lab, we're going to be talking about patterns on small spatial scales, but you can also look at them on large spatial scales. So I did another video on that. This is an example from that video where we're looking at across North America the distribution of birds, and we can see that they are very clumped, that you have areas where you have very high density populations and then wide areas where there's low densities of the populations. In the top one, we're looking at the American crow. And then down here, we're looking at the fish crow, which has a few hot spots where you find lots of the population. We're going to be looking at plants in a lawn, looking at lawn weeds and things that you're going to need. First of all, you're going to read over the safety information for your lab and make sure that you are not exposing yourself to any unnecessary hazards. In our lab kits, we are going to be using both of our measuring tapes, the 30-meter tape and the 3-meter tape. The 30-meter tape just to make sure you have a big enough area to work in. We're going to use the 4 metal stakes and the twine, and that's how we're going to set up our plots. We're going to need to take a charged phone with you. Make sure you have a weather app so that you've got the notifications turned on just in case some lightning is coming your way. And you're going to need to be using the camera in that. You might also find it easy to use it for looking up the plants and make sure that you've got the right identifications. You're going to find a location, so you're going to find a lawn area with weeds, and you're going to find a safe place with at least 2 different types of weeds that you can identify. And make sure that it's at least 50 meters by 50 meters in size so that you've got enough places for your samples. And then you're going to need the lab book. With your lab book, bring pencil. Don't forget your waters, your sunblock, your sunglasses, your hat. Make sure that you're dressed appropriately for the weather. Depending on where you are living, there's probably different weeds that you find commonly in the lawns in your area. So wander around the park, see what you can find. These ones are common in our area in South Jersey. There's the nice little white clovers, which kind of have a V shape on their leaflets. These are 3 leaflets that make up their leaves. And make sure that you can tell the plant just from the leaves, because they won't often have the flowers on them. So when you have identified something, take some samples of the leaves and keep them with you, and that way you'll be able to recognize them again. So there's a couple of plantains around here, like a broadleaf plantain and a buckhorn or a narrow-leafed plantain. There's dandelions, which there's other things that kind of look like dandelions. If you break the leaf of the dandelion open, you'll see it has white sap that comes out of it. So make sure that you can identify at least 2 species, and you're going to be sampling those. And put down the name of the species and photograph each of them. My students are putting together a blog which will have a photograph of their species names, and that way we can see if 2 different people are calling the same species different things. So once you found your study site, make sure that it's big enough and that you take some pictures of it, because you're going to have to be able to describe that study site. Is it a sort of a shadowy area along the edge of a forest? Is it an open, bright, sunny park? Does it have a lot of people in? Is it an isolated area? Is it you're in your backyard? So make sure that you can describe what that study site is like. Select your 2 species, be able to recognize the leaves and take pictures of them. And then we're going to be doing random plot samples. So you're going to be doing 2 different sizes of plots that you're going to be looking at the 2 different species in. You're going to be doing plots that are 20 by 20 centimeters in size, so that's a square plot. And then you're going to do a rectangular plot known as a Meyer plot, which is going to be 20 by 50 centimeters. You're going to do 25 of each. In each one of those 50 plots, we've got 25 square ones, 25 rectangular ones. You're going to be looking at the numbers of the 2 species that you're interested in. And I want you to take a photo of 1 plot of each size when you set them up. They take a little bit of time to set each one up, because you've got to put down your stakes and measure it out. And then, so take a photo of a couple of them so that we get an idea as to what you're looking at. So here's a photo of 1 plot that I have that was 20 by 20 centimeters. So I took the photo. You can see the little metal stakes from our lab kits are sticking on in the corners. It took a little bit to get kind of right angles here, just by measuring it. Then I took my twine and I just wrapped it around and tied it at one end so I can see where the edge of my plot is. And you're going to record the cover classes. So let's just say I'm looking at these clovers, these white clovers. In order to figure out the cover class for the clovers, well, it's not 0, there's more than 0, but make sure if it's 0, you record 0. So let's say I was looking at dandelions in here, I would put dandelions at a 0. Cover class for clovers, if it looks like if I took all these clovers that are in here and if I squeeze them all together, is it going to be more than 1/4 of the plot? If it is, and if it's less than half the plot, then I'm in cover class number 2. If it's more than 3/4 of the plot, if it's like covered with mainly clover, then you know it's cover class 4. So you're going to be recording these in your data book. And make sure you record the zeros, because the zeros are just as important to know a species isn't there than if it is there. So you're going to just have to take some time. When you're doing this, if there's a species, it might be an individual is rooted out here, but its leaves are in your plot, we're going to count them. So anything that has leaves within your plot, you're going to actually count them. So we're going to be using these cover classes. Later, when we're doing calculations, we're going to convert these cover classes to the midpoints of those ranges to do the calculations. The 20 by 50 centimeter plot is known as the Meyer plot. And this plot was figured out to be the most efficient way of sampling this type of vegetation. Using a plot this size gets you the most accurate information for doing this kind of study for this type of vegetation. So these are herbaceous plants. These are plants that die back to the surface every winter, like the grass, the clovers, the dandelions. Some of them are perennials, and their roots will just live over the winter and they'll come back from the roots. Some are going to be annuals that are going to come back from seeds every spring. Anyway, once again, you're going to place your 4 stakes, 20 centimeters wide by 50 centimeters long. Wrap the string around the plots, take a picture of one of them, and then you're going to, once again, look at the plants in here. So in this case, I do have some dandelions and I do have some clovers, and I have to do the cover classes again. So I have to figure out if I take all these clovers, do they even get up to 25%? Is that a cover class 1? If the dandelions, it looks like there's quite a bit more. Is that a cover class 2 or 3? So try to figure it out if you trying to see what percentage of the plot. This is a modified Meyer scale. There are actually more categories. There's 2 more categories. There's like they squeeze in a smaller one and a higher one. I just thought it was easier for you if you just got an idea how to use this. Also a reminder, use the zeros. Record the zeros in your data sheet. So if you did a plot and there's no clover in it, put a 0 for the clover, and you can look at how many dandelions there are in it. So in order to determine your locations, you're going to determine these at random. So what I want you to do for getting a random location is to come up with a system where you're going to pick random spots. So you could take a frisbee and throw a frisbee out. You could take a stick and throw a stick. You could throw a ball. You could just take something that you're going to randomly throw in your area, and wherever it lands, that's where your plot's going to be. So here I have 3 random spots that were chosen. The yellow is going to be the 20 by 20 centimeter plot on there, and then the 20 by 50 centimeter plot. Obviously, these are not to scale, but you get an idea that you're going to be putting up 25 random points, and at these points, you're going to do the 2 plots. And each plot, you're going to do 2 species. So you're going to get 1 species in the square plot and the rectangular plot, and a second species in the same square plot and the regular rectangular plot. You're going to do 25 of these, of each of these. So our data sheet in the lab book looks like this. It's got the little cover classes so you can keep track of that. Put the names of your species, describe your location, the temperature, the time of day, what the weather's like, the date, and then in each one of these squares, you can put one of these cover classes. So you could write for species 1 in plot 1, it was cover class 2, and in plot 2, it was cover class 1. This is the 20 by 20 centimeter plots. Put in the cover classes for species 2. And down here, we do the same thing. When you do the Meyer plots, you're going to end up with filling every one of these boxes with the number of each species that corresponds to its cover class. So you're going to do the 2 types of plots, the 20 by 20 squares and the 20 by 50 Meyer plots. You're going to do 2 species in each one. Don't forget to include your zeros. And your sample size is going to be 25 plots of each of them. Once you've got your data all together and you've got all 50 plots sampled for both species, and you filled in all your little zeros and 1s and 5s and zeros and 0s and 5s, whatever it is in your lab book, you're going to then submit it to for my class on the data form. You'll do a separate data form for each species in each plot. So each row of your data sheet is a separate form. So that's the species 1 on the square plots and species 1 on the Meyer plots, species 2 on the square plots and species 2 on the Meyer plots. And then you're going to update everything on your Ecology lab blog. The next step is to do the calculations, and so you're going to set up a data table. So open up Excel and you're going to make a spreadsheet for each of those 4 data sets, 1 for each species on each plot. By the way, I put wolves in here because they're a clump distribution, because you can get a little pack of wolves in this valley and a pack of wolves in that valley with spaces between them without any wolves because they need the big hunting grounds. Once you've got your data into a data table, we're going to be converting the cover classes to the midpoints of the cover classes. So the 0, 1, 2, 3, 4 are going to be going to the midpoints. And then we're going to use the midpoints to calculate the mean and variance of each data set. And we're going to include the zeros in that, so we're not ignoring the zeros. And from that mean and variance, we're going to take the ratio, the variance to mean ratio, in order to calculate the dispersion index. So I'm going to just explain that a little bit. Here we have 3 scenarios of individuals of a species scattered in different patterns. So the pattern on the left is going to be where we have a uniform or regular distribution. The middle pattern is a random distribution. So there some are close to each other and some are further apart from each other. And then the last one where we have these little clustered patterns, that is going to be a clumped or a contagious distribution. And the little squares, the little yellow squares, are representing your 20 by 20 centimeter plots. When we spread them out and sample the individuals in the plots on the uniform distribution, we find that we're pretty much getting the same number of individuals in every plot, 1 or 2. So there's very little difference between plots. The statistic we use to measure distance is going to be a measure called variance. And we show that with the symbol S squared. S is the standard deviation. So variance is the standard deviation squared. And it's going to have a low variance. They're all going to be pretty much the same. So there's very little variability. We measure variance as how different from the average each plot is. And they're not going to be very different. It's going to be a very low number. This one over here where it's clumped on the right-hand side, some of these have zeros, some of them have tons. There's very high variance. So on that one, we have high variability. And then in the random one, we've got a little bit of everything. And so we end up in the random one, we have medium variance. The measure of this pattern, dispersion pattern, is quite simple to do then. We just take that variance and look at it in relation to the mean. If you have a random pattern, the variance is equal to the average. So what we do is that we take the ratio, we calculate the variance, and we calculate the mean of the numbers that you took from that area. And if it's close to 1, then we know the pattern is random. On the left side, remember the standard deviation, or the variance is low. So if we take a small number and divide it by the mean, it's going to be smaller than the mean, and so that's going to be less than 1. So if you get a little number, you know it was a uniform pattern. And then on the right-hand side, we're going to take the variance divided by the mean, and if it's greater than 1, then we know it's more clumped than it is random. So it's leaning towards the clump side if it's greater than 1. And if it's near 1, it's random. And if it's less than 1, it is more towards the uniform or regular side. So we're using the textbook Ecology, Concepts and Applications. And at the end of the 8th edition, they've got these sections called Investigating the Evidence. And number 3 tells you how to calculate the mean. And when we're calculating the mean, we're going to be using the midpoints of the cover classes. Remember, we were using those cover classes. Then we'll be calculating the mean or the average. Investigating the evidence, number 4, talks about how to calculate variance. So what you do to calculate variance is you see how different every value is from the mean, and you can add up all those differences. And we square them just to take care of the negative numbers. And then the last thing you're going to do is you're going to take that variance, you're going to divide it by the mean to come up with that measure of dispersion. And when we look at the relationship between the variance to the mean of those different distributions, we find that the variance is greater than the mean, which in a clump distribution, so there's a lot of variety, which means the variance to mean ratio is greater than 1. And that's explained in investigating the evidence 4. So number 3 talks about the mean, 4 talks about the variation in the data, and 9 goes over this clumped random and regular distribution that we've already talked about. Your first step then is going to be putting your data, just a long column of numbers for your cover classes, into Excel. So I make a separate sheet for each species in each plot. So I make up 4 different sheets, and it's just going to be the list of the cover classes. And then we're going to, you can set your table up because we're going to then be calculating the midpoint of those, which we have a little table to refer to. We're going to figure out how different each midpoint is from the mean, from the average. So we have to calculate the average of all of these, find the differences, square those differences, add them all up, and then we're going to divide by the sample size minus 1 to get the variance. Then we can take the variance to mean ratio, and we'll be able to get the variance to mean ratio of our different data sets. So remember, we have 4 different data sets. So I have already calculated the dispersion index at variance to mean ratios for 3 of these data sets. I've left one of them. I'm going to do that now on my example data set to show you how to do it in Excel. I'm sharing with you my screen as I'm working in an Excel data sheet. So I made an Excel sheet, Excel data set, and I made 4 different sheets in it. So I'm in the one that's called Clover Square. So I've named my sheets down here at the bottom. So I've got Clover Square down here. And then there's Dandelion Square. So I have a data set with clovers and dandelions in the square plots. And then if I move over here, I've got the clovers and dandelions in 2 separate data sets in the Dublin Meyer plots. And then I've made a 5th sheet, which is just going to be putting my summary data when I'm all done. So right here, I've set up my columns. I have the cover class, the midpoint. Remember when we were doing our cover classes, they were for a range of values. And so I have just made myself a little table here, which I've highlighted in yellow, which is showing you for each cover class what its midpoint is. And that is out of our lab manual. And then, so I'm going to be showing you how to use something called VLOOKUP in Excel to actually read off this table. So I've typed in here my list of 25, and it says row 26 because the first one is my column headers. I've typed in my 25 cover values for my clovers in the square plots. And I'm going to show you how you can convert that to these midpoints. Some of the other things we're going to be using in Excel, we're going to use sum, I'm going to show you the average function. We're going to be using this name box up here again, like we did in a previous lab. I'll show you how to square things. I'm also going to show you the var.s function, and we're going to use this value lookup, this VLOOKUP. So the very first thing we have to do is convert our data. So the way we're going to do that, we're going to just use a function. When we're going to use a function, you type 0, and we're going to type in the function name, VLOOKUP. And then it wants to know, what is it that I want to look up the value for? Well, I want to look up the value for this cover class over here. So I'm just going to click on that square. So that's square A2. So I'm going to look that one up. Where am I going to look it up from? Well, that's going to be from this little table I typed in. And I'm going to highlight the squares in that little table, which is going from E2 to F6. And when I want to be able to drag this all down to all the other rows when I'm done. So I just want to make sure it always goes to the right place. So I'm just going to make sure it goes exactly to those spots. So I'm just adding in my little dollar signs. And then it says, okay, let's say I found the value 2. What do you want me to put in the midpoint there? And I want you to read from the second column in this table. So I put the number 2, the second column of the table, and hit enter. I'll close my bracket first. So let me just go over this. VLOOKUP, open bracket, then the value that I'm looking up, which is that A2, the table I'm going to be looking at for it in is over here. And then what column in that table will actually give me the answer? It should come up with the answer 37.4 for the value number 2 here. So let's hit enter, and it did it, 37.4. So if I drag this down by the little box in the corner, for 0, it found 0. For 1, it found 12.5, and it could just go and read all those values. And that saves me having to look them all up and type them in there. So it saves you a bit of time. So now we have the midpoints. These are the values we're going to be using for our calculations. The very first thing we're going to do is we're going to total that column. So we're going to get the sum. So just equals sum. I'm going to highlight the numbers I'm adding together, close my bracket, and hit enter. And I have 649 from all those numbers added up. My sample size is 25, right? I know I have 25 samples that we took, 25 plots, so I'll put that there. And then if I want to calculate the average, I take the sum and divide it by the sample size. So the average equals the sum, 649. I'm just clicking on the 649, divided by, and then I'm clicking on 25 and hit enter. Now that sum I'm going to be using again because I'm going to see how different each one, or sorry, that average, each one of these is from the average. So that thing that sum divided by n is actually the mean, right? It's the sum divided by n, the sample size. I'm going to name it. So up in this name box, I'm going to give it a name. I don't want to use mean or average because those are functions, but I want to say something about the sample because there'll be a different one for each of these. So I want it to be like the clover square mean. So I guess I could call it like CL for clover, SQ for square, and mean. No spaces in those. And once I have named it, I click enter. And now if I click on that box, that name will pop up. So that's going to be a constant. So I've named that as a constant. Now, there is a function in Excel to calculate the average. In the past, and sometimes when you make mistakes in how you're doing things with the sample sizes or not reading the right data, you might want to double check things. So what I'm going to do is just use that average function to make sure to double check my calculations. So I'm going to say average equals average, open the parentheses, highlight the data set, close the parentheses and hit enter, and look, they match. I'm so happy. Okay, so that's good. So the next column is how different each one of these is from the mean. So all I have to do is go equals 37.4 minus, remember what we called the mean? It was CL SQ mean, and hit enter. And so I get the difference from the mean. I can just drag that down because the mean is a constant. It's going to work. So no matter where I click on here, it's just doing, now it's what's in B25 minus that constant, or what's in B23 minus that constant. So it keeps going back to that constant because I named it. So the name box is your friend. So you can see there's negative numbers and positive numbers. And if I added all those up together, they'll balance each other out, the negatives and the positives. But I want to find out sort of what the overall difference from the mean is, how much variability there is in the data. So in order to do that, we square it. So we're going to go equals click on the value, and then if you're going to take the power of something in Excel, you use a little caret. It's on your keyboard, usually Shift 6, and then I can put a 2 for squaring. And then I can go ahead and drag that all the way down. So all of those now, so those negative numbers have now become positive numbers. And because my original data over here had 1 decimal point, I'm going to keep 2 decimal points on my data, but I don't need 4 decimal points sitting here. To calculate the variance, I'm going to have to sum all of that. So it's going to be sum of that whole list, big number. And then I'm going to get the variance, I'm going to take that number and divide it by, I'm going to open a bracket, the sample size minus 1. That's because we've lost a degree of freedom, statistically speaking. So because it's not the whole population, we don't divide it by the actual sample size, we lose one of those samples and it's minus 1. So we've got the sample size minus 1, which would be 24. So we're going to take that big number and divide it by 24. And that's our variance. Now there is a particular function called VarS that calculates variance of a sample, which is different than variance of the actual whole population. So just to double check that we've done this right, I'm going to use that. I'm going to just try it out. So equals var.s, and then I go to my original data, enter, and look, they match up. It's perfect. So the very last step is to get the dispersion index, which remember is the variance divided by the mean. So it's going to equal the variance divided by the mean. And here it's quite a bit above 1, so we can interpret that. So I've gone ahead and I've done that for the dandelions in that plot and got the data, and the clovers in the Meyer square, and the dandelions in the Meyer square. And then this last page, I made a little table. So these are the 2 plants. These are the different sizes of plots. And then I put the means and variances I calculated in those. And so the plot that I just did, the mean that I calculated, I go equals, go to my data set, and type in that mean. The variance I calculated, go equals, go there, and just click on the variance. It puts it in. And the dispersion index equals, go over there, click on the data set, click on the dispersion index. And let's put it all in there. Now I made this little graph over here. All I did was I selected out these first 2 columns and that last column, and I went insert, column graph, and there, you get it, right? Move that around, you can make it bigger or smaller, adjust it. So I could do the same thing if I wanted a graph of the mean and the variance. I could just highlight that, insert, column graph. Now, mean and variance are on very different scales, but you can see in all cases here the variance was much higher than the mean. So you can play around with your data, and then you take a look at your data, and you might come up with some ideas of some patterns. So that's how to use Excel to calculate a dispersion index. So when you have all your data, you're going to be able to see whether your patterns are close to random, close to clumped, or close to regular. You can also look at differences between the patterns in your different samples, and maybe you can come up and see some sort of ideas or questions or hypotheses that you could test using these data. My students will update their lab blogs, so they will add into their lab blog the graph that they've produced of the dispersion index, but also the data, not the raw data, but just the means and variances that they calculated for each data set, and the dispersion index that you calculated for each data set. And then on eLearning, I have a little assignment for you to submit some of your information. Thank you so much for joining us on this. I hope you can understand how to calculate dispersion index now. You learned some tricks in Excel along the way, and I'm going to put the link for you to click on here so that you can look at the video on patterns of species distributions on small scales, where we talk about the different types of dispersion patterns and also interpreting them at large spatial scales as well. So hopefully you find those useful.